Want to start recording again?
Yes,
Okay,
please.
perfect. Okay, thank you. Okay, so I'm going to just move on and show you guys just very briefly what this rule is really capable of doing, and then I'll move on to introducing spike time independent plasticity. So this rule was initially motivated because the authors were studying how this selectivity can arise in the visual cortex. So they were looking at selectivity in of single neurons in the visual cortex. So you probably know from the classical Hubel and Wiesel experiments that these neurons are indeed orientation selective. And we know that early in development, they're not. And so somehow there's some input. So if you show different input patterns that contain signatures of these orientation selective inputs, then the postsynaptic neuron in the cortex will become selective to one of these orientations. So here, let's say the postsynaptic neurons, apologies for the change in notation. Here I use Y as the postsynaptic activity and X as the presynaptic inputs. So the setting in which they applied this BCM rule was a scenario in which the postsynaptic neuron received inputs from basically an entire array of neurons, where different groups of input neurons basically code for a different orientation. So in this case, we assume that inputs 1 and 2 code, for example, for this orientation, 45 degrees, inputs 3 and 4 code for the vertical orientation, inputs 5 and 6 correspond to the minus 45 degrees, and so on. This is just a schematic. Of course, we have a scenario where more than 2 neurons code for a given orientation, and also older orientations are represented. So this is just a schematic. But the idea is that the postsynaptic neuron, at least in the network, receives a stimulation that corresponds to only one orientation, but that stimulation changes at different time points. So at time t1, let's say the postsynaptic neuron receives the stimulation that corresponds to the vertical orientation. It's sort of like showing the animal a grated bar, or grating, or an oriented bar with the vertical orientation. Then at the next time point, 100 milliseconds later, you show a different orientation, and so on. I call this dynamic input presentation because it means that at different time point, you change which orientation you show, meaning you change which group of neurons you stimulate, where a given group of neurons codes for a given orientation. And so in the steady state, this rule, as Venus, Cooper, and Monroe showed, this guarantees the scenario where one of the orientations will become selected, meaning that only one subset of the connections, in this case, the ones that correspond to inputs 5 and 6 and the orientation minus 45 degrees, will become potentiated, and all the other ones will become very weak. I've denoted here by the thicker arrows, and then the thinner arrows denote very weak connections. And so that's basically what they showed. So you have to look at the book to really understand the mathematics for why this particular rule enables this to happen. And this matches, of course, nicely what Hubel and Wiesel showed. So this is actually a model, but a model curve, but you represent all the orientations, and then you can measure the response of the postsynaptic neuron to all of these different orientations. And so here we saw that the neuron becomes selective to this minus 45 degrees orientation, meaning that the connections from that set of neurons that encode that orientation become potentiated, and therefore the response of that neuron is maximal for that orientation, and then it decreases as you change that particular orientation. So this is what the rule can do. This is different than, let's say, the covariance rule. So this self-normalizing aspect, this adjusting of the threshold between LTP and LTD, enables the rule to achieve this. So I'm now going to shift gears a little bit, and I'm going to show you now models of synaptic plasticity that use a different type of activity pattern than the firing rates. Everything we did so far was this Hebbian rule, so either the correlation or the covariance rule, and the BCM rule, which explain or model synaptic plasticity as a function of the activity of the presynaptic neurons, where the activity is measured by the firing rates. But nothing in the Hebbian formulation says that it should be firing rates. And in fact, Chuen asked this question at the beginning of the lecture: what is a realistic pattern of inputs that these neurons experience? And of course, it's action potential. So in recent years, and in a moment we'll define spike time independent plasticity, people have shown that different patterns of spikes can change the synaptic connections. And this has led to the definition of spike time independent plasticity. So the idea is that you apply different protocols. So you take your presynaptic, your postsynaptic neuron, you stimulate them with different patterns of inputs, in this case, spikes. And then you measure, similar to what we showed before, for rate-based plasticity, the synaptic connection, either by measuring the postsynaptic current or the postsynaptic potential. So what are the types of patterns that people use to evoke synaptic plasticity here? So the very classical experiments done by B and Pu in 98 applied spike pairs. So one spike evoked in the presynaptic cell and another spike evoked in the postsynaptic cell that were separated by a timing delta t. And then they repeated these pre-post pairs multiple times. I don't know, about 60 pulses. They were presented at a given frequency. And then they characterized the amount of change in the synaptic connection upon the presentation of these spike pairs. They did this by presenting first the pre and the postsynaptic spike and changing this timing delta t. And they also reversed the order of the spike. So first they showed the post and then the presynaptic spike. So this gives rise to this spike time independent plasticity curve. I'm sure you've all seen this in some context. So this is probably not a surprise. And what this particular plasticity rule shows us is what the amount of synaptic change is, as indicated here on the y-axis, as a function of the timing between these pairs of spikes. So if the timing is positive, meaning you have the occurrence of the pre and then followed by the postsynaptic spike, the synaptic connection will be potentiated. And otherwise, if you have post followed by pre, you have synaptic depression. And depending on how big this timing is, the effect gets reduced. So this is how we can formalize this rule mathematically. We can define A plus here and A minus to be the maximum amount of potentiation and depression. You can see if the timing that will be observed, if the timing between the spikes delta t is close to 0. And then the effects decay. And so one can fit these exponential kernels with two time constants, tau plus, to denote how fast this potentiation decays as the timing between the pairs increases, and tau minus to indicate how much the depression decays if the timing between the two post followed by pre-spike decreases. Now, STDP, this is kind of interesting, does not require these separate normalization schemes that I showed you before. If you ensure that what people call this depression to potentiation ratio, so the product of the A minus and tau minus divided by A plus and tau plus is bigger than 1. So this is actually a result from Song and Abbott 2000 and 2001. So that's spike timing dependent plasticity. But even then, the story is not so simple, because after this initial discovery of STDP, people started investigating various aspects of the rule. So let me just highlight a few of them. People found that actually the shape of this STDP function depends very much on the dendritic location, right? So I showed you this slide here that classically people inject spikes in the soma, but if instead you end up injecting spikes in the dendritic tree, how far you do that from the soma, whether you do it in the apical or the basal dendrite, can really have an effect on what you observe. There's a nice experiment from Rob Fromke and Yang Dan from Nature, 2006 maybe, where they investigated exactly this phenomenon. Moreover, the spike patterns are not just these beautiful pairs of spikes shown always in this regular structure, but they can have complex activity patterns. Notice that in most of the rules that people have described in different brain areas, you see this longer depression time scale than the potentiation time scale, but this is not always the case. You could have different ways in which the spike pairs can interact. So is it just neighboring spikes that interact, or all the spikes in your spike train? So these are all differences, and people found that basically in different brain regions, different things hold. Another characteristic feature is the temporal symmetry. And so people find that if you apply different kinds of neuromodulators, you could actually change the shape of the STDP curve and actually have a symmetric function, and so on and so forth. So there's a lot of things that can be, you can modify the rule and change the shape and change the effects that are observed with this STDP function. I will address the frequency dependence in a few slides, so you can keep this one in mind. And this leads me to the question that we got asked at the very beginning. So how should STDP, or this rate-based plasticity that I described before, but especially STDP, how should it be applied to naturally occurring spike trains? So, in adult or in development, what really goes on? Are these rules really operating under the hood, or is there some other more complex learning rule that uses these more naturalistic spike patterns? So the majority of the STDP rules have used these classical pairing protocols, but why should it be that pairs of spikes should be the elementary building block of synaptic plasticity? So very briefly, I want to show you an example of the early visual system where I tried applying STDP and actually did not succeed. So this was actually my very first paper as a PhD student. So it was published some time ago in 2009. Where I was looking at the refinements of synaptic connections in the developing visual system in the thalamus, in particular in the visual area of the thalamus known as the lateral geniculate nucleus. And I was looking at how projections from retinal ganglion cells in the lateral geniculate nucleus get refined. I was especially interested in naturally occurring patterns in early development that occur in the form of retinal waves. So this is the period when the eyes of the animal are closed. So the animals cannot see anything, but yet they still experience patterns of neural activity that have the shape of retinal waves. And this is what the retinal waves look like. So here we see completely spontaneous activity. This is not sensory evoked. Each of the little dots that you see are electrodes in a multi-electrode array. This electrode was put in a dish together with a retina that was extracted from the young animal. And then what you see with the big circles are activity patterns that were recorded for each of these electrode sites. So the size of the circle denotes the amplitude of activity, so the amount of firing that was recorded at that array. So in this case, we were interested in looking at refinements between the retina and the lateral geniculate nucleus. So that's the network we have in mind. And this is the simulations, the framework, the modeling framework that we have. It's exactly the same feed-forward network that we've studied before. So we have neurons in the retina. So in the back of the retina, these are the ones that are being recorded that experience these retinal wave patterns. And then we're looking at how this neuron in the thalamus refines its connectivity in a developmental setting driven by types of plasticity rules such as STDP that we characterized, and by these natural activity patterns in the form of these retinal waves. So what do these activity patterns look like in these retinal waves? There was a study shortly as I was starting my PhD that got published that characterized these activity patterns in two types of ganglion cells known as on and off. So on and off in the adult animal that can see are neurons that respond to light onset and light offset. So if the neuron likes to fire when you trigger the light on, that's called an on neuron. But if it fires when you switch off the light, that's called an off neuron. But in development, you don't have light, and yet these neurons are still there. So they're kind of getting prepared, they're being wired up to prepare the animal for adulthood. And during this period of retinal waves, the experimentalists found a particular pattern of firing. So I hope you can see here in the zoom, where the off cells generally fire with slightly longer bursts, but importantly, their firing is temporally delayed relative to the firing of the on cells. So you see that they fire about 1 second later than the on cells. So to determine exactly quantitatively what this delay in firing is, the experimentalists computed the Pearson pairwise correlation coefficient. So this is shown here at the bottom. and averaged it across specific pairs of cells. So the middle is the cross pairing, so the same pairing on and off. And then here in the purple and in the red, they did the cross pairing. So if we look at the purple curve, this shows us the pairing for on-off pairs, so pairs of different kind, and you see an offset of this peak at -1 second that is basically giving us a way to quantify what we see here in the spike trains, that the firing of these off bursts is offset, so it's 1 second later than the firing of the on bursts. So we do the same modeling approach that we showed before. We have a postsynaptic neuron in the in this case in the lateral geniculate nucleus that receives these on and off inputs. And what we're interested in is coming up with a learning rule that will tell us how to describe the change in the synaptic weights. So here we write the differential equation as before. So what's the change in the weight after a time step delta t? Here I use W as the learning rule. Previously I had used F, but it's the same thing. It's some unknown function of the presynaptic activity denoted here by X and the postsynaptic activity in the LGN denoted by Y. But now what is key here, rather than showing artificial simulated activity patterns, which a lot of the time we as computational neuroscientists do, we took real spike trains here for the X's that were recorded in the retina during these retinal waves of the on and the off cells. And so this was the time when STDP was very popular. And so I said to my supervisor, I'm going to take this data and I'm going to plug in here the STDP rule, and I'm going to see how subject to this STDP rule, how these activity patterns influenced or driven by these retinal waves drive activity in the postsynaptic neuron. Now you may ask, did I have or know the activity of the postsynaptic neuron? The answer is no, but I modeled it. Here I built different models. I had a leaky integrate and fire model. I had a bursting Isikievich neuron type of model. It didn't matter. I was focusing primarily on the plasticity rule, and I took at first an STDP rule. And what I tried to quantify was how this postsynaptic neuron, how selective this postsynaptic neuron becomes to just the on or just the off inputs. Because we know that as the animal develops, the LGN neuron goes from being weakly connected to both types of neurons to being strongly connected to only one type of neuron, namely becoming only on versus only off. So we're going to model how selective this neuron is by calculating what I call a segregation index. So we have a segregation index of 1 if the neuron becomes only selective to the on inputs, meaning that only the weights from these on inputs become strong and the others become weak. And the neuron gets the segregation index of -1 if it becomes selective only to the off inputs. So only these connections from the off neurons become strong. So we use the STDP rule, and you see that I found segregation indices all over the place for the 6 data sets that I acquired from the experimentalists. So I concluded that no matter which type of model I used for the postsynaptic activity or how I adjusted the STDP rule, I was fixing the time constants, this potentiation and depression, I could never improve the results. So why is that? So let's take a careful look both at the activity patterns that are driving the postsynaptic neurons, so from the retina, and the learning rule. So notice that the learning rule can evoke potentiation or depression by basically pairing that depends on a delta t that's on the order of tens of milliseconds. So already when you reach timings of 80 milliseconds, where the pre is followed by the postsynaptic spike is longer than 80 milliseconds, you basically don't see any synaptic change. So you need to separate your spikes by very short time windows in order to see potentiation or depression. But if you look at the activity patterns in the retina, especially these cross-correlograms, this Pearson correlation coefficient, you see these are offsets of 1 second. So these are, the scale bar here is 2 seconds. So yes, you have spikes, and yes, they occur in short bouts of time, but the relevant correlations that exist in these activity patterns are significantly longer, seconds, as opposed to few milliseconds in the plasticity rule. So we effectively have a mismatch. We're trying to apply a learning rule that's basically operating on such fast timescales relative to the timescales that are evoking or driving the postsynaptic neuron that effectively this rule is trying to just amplify gibberish, right? So it's doing nonsense. But luckily for me at the time, there was a recent paper at the time that identified a learning rule and measured it exactly in the same pathway that I was studying from the retina to the LGN. And they did this in the same developmental setting that I was studying. So just before eye opening, when the animals are young. And what they found is rather than the STDP rule that I have been showing you so far, that operates on these short scales and has this asymmetric window of potentiation and depression depending on which spike comes first, they actually describe the learning rule that depends on these very long time scales, just like the activity patterns we saw in the retina. So here on the y-axis, again, they show the change in the synaptic weight, characterized experimentally by change in the postsynaptic current. And on the x-axis is shown the timing between pre and postsynaptic activity. So rather than spikes, they used bursts because they noticed that in development, most of the activity that occurs in these retinal waves is indeed driven by bursts. And so as a function of the timing between the pre and the postsynaptic burst, very similar to STDP, except now we use bursts and the start of the burst is what determines the timing. They found that now the new learning rule operates on these long time scales. So the x-axis here, we see 1,000 seconds, not a few milliseconds. Sorry, 1,000 milliseconds, which is 1 second rather than a few milliseconds. And the second difference, in addition to the time scale relative to STDP, is the symmetry of the rule, right? So STDP is asymmetric, meaning that it matters whether pre comes before postsynaptic activity for potentiation versus depression. This new rule that they called burst timing dependent plasticity, or BTDP, does not care whether the pre or the postsynaptic burst comes first. So as long as they're separated by a short amount of time, either one can come first and then you have potentiation. And if they're separated by a long time, then you have depression. So we can describe this learning rule in the following way, just like we had the equivalent for the STDP rule. So we have a single A+, we have the tau plus here, and then we have the depression if the bursts are separated by more than 1 second. So now we actually have a match between the timescales in the learning rule, which are on the order of a second, and the timescales in the retinal waves, which are also on the order of a second, these delays that we saw between the on and the off cells. And when we apply this rule in our network, we've actually find that in the 6 different data sets that we have, reliably in about half of them, we can achieve so-called on selectivity, meaning the postsynaptic cell only strengthens the connections to the on neurons in the retina. And in the other half data sets, the neuron in the LGN becomes selective to only the off input. So it becomes off selective because now the segregation index is -1. So we have this very robust selectivity that is driven by this burst time independent plasticity rule, where the time scales match to those of the activity patterns. And I'm going to now go back to the STDP rule, because in addition to just caring about development, we want to explain plasticity overall. But I hope you keep in mind this lesson, that the plasticity rule that you choose to put in your networks should match the timescales of the activity that you know is endogenous to the system that you're studying. So if you know that your system likes to burst, even in adulthood, right, then you should be using learning rules that use bursts. So I go back to STDP because there is also a way to elongate the timescales in STDP. And the way to elongate the timescales in STDP is to actually assume that it's not just pairs of spikes that evoke potentiations and depression, but it's actually the addition of other spikes, for example, triplets. So you add a third spike and you go from pairs to triplets that now evoke potentiation and depression as a model to kind of elongate the timescales. So I'm going to explain to you in a moment this new triplet spike timing dependent plasticity rule. And I'm going to start by telling you how experimentalists discovered that this could be a way in which plasticity operates in sensory systems as well. So they started with the classical STDP rule of B and POO. They started with this pairing of pre and post spikes. And they observed that the behavior that comes up when you increase the frequency of pairing. So you still show the same 60 pairs, but you now all of a sudden show them in a shorter span of time. So you increase their frequency. So they saw that when you increase the frequency, you get the somewhat paradoxical behavior. So if we do a thought experiment, and if we say, hey, we have pre-post, and we know that post causes potentiation according to DP. Now we're going to increase the frequency. So we're going to have LP potentiation whenever pre comes before post. But remember, if we now introduce the new pre-post pair, we have depression because we now have the occurrence of a post-pre spike, right? Then we have the next pre-post and so on. And so what this would effectively give you is a scenario where every time you have potentiation or LTP because the pre-post occurred, because you show the next pre-post so close in time, you've increased the frequency of presentation, you will have this potentiation be canceled by the depression of the post-pre. So if you then add up the contributions, pre-post is now canceled by post-pre, you have a new pre-post canceled by the next post-pre, you will effectively see no change in the synaptic connection, or you expect to see no change if you just increase the pre-post frequency. But if you actually look at the data, that is not what happens. So let me unpack this plot a little bit. What we show here on the y-axis is the change in the synaptic weight, and on the x-axis is exactly the frequency of the presentation of these pre-post spikes. So let's look at the experiments which are shown in the black full line. So this is the black full line where we increase the frequency. So if we increase the frequency, what they see rather than no change, which is what our model would predict, as we explained, because every pre-post is canceled by a post-pre spike, you actually see that as the frequency increases, the amount of potentiation increases. So you no longer see flat change. So if we now look at the comparison of the model, this is the full blue line, you see that if you increase the frequency, the model will predict effectively no change. So at the frequency of 50 hertz, you see that the delta W is at 0. But in the experiments, that's actually positive. So even though we predict this no change, because the post-pre cancels the pre-post, we see a potentiation. And same if you now do the opposite pairing. So if you show the first the post and then the pre, this is shown in the dashed curves. Again, we have a mismatch between the actual recordings shown here in the dashed black line and the prediction from the classical pair-based STDP model, which is shown here with the dashed blue line. So instead, Jean-Pascal Pfister and Wolfram Gerstner had this proposal that maybe a third spike should be introduced to the model of STDP. And so I worked with Jean-Pascal eventually to analyze the mathematical properties of the rule. So what the rule says is that you add a third spike so that instead of just the timing between the pre and the post spike denoted here by delta T1 mattering for how much potentiation and depression you induce, you also use the timing between the two postsynaptic spikes denoted here by delta T2. So this is the triplet now. So if that timing is very small, so if this delta 2 is small, you evoke some potentiation. But then if you increase the timing, the amount of potentiation effectively drops. And so if that third spike occurred very, very far into the past, then you will not influence the amount of potentiation at all. You can do exactly the same thing by introducing a second presynaptic spike in your triplet. And then you can modulate the LTB window of your classical pair-based STDP rule. But Jean-Pascal Pfister and Wolfram Gerstner, when they proposed this triplet model, they actually found that you can describe most of the data in the hippocampus and the visual cortex where these experiments of the changing frequency were done by just adding a third spike for the potentiation. So they found that the depression can be basically described primarily by the pair-based rule. So I hope it's clear now. We introduced a triplet model by adding a third spike that modulates the amount of potentiation that is classically described by the pair-based STDP rule, where now the curve for the STDP rule is determined by the timing between the two postsynaptic spikes. And the way to describe this mathematically, so I'm only doing this for the potentiation part. You can also do it, of course, for the depression part. For the potentiation part, you see that now instead of the single exponential that we had for the classical pair-based STDP rule, we have a second exponential. So we have two delta t's, one delta t1 and delta t2. The first one is exactly the same as in the pair rule, but now we have a second one with a time scale tau y. And the key here is while tau plus was operating on this time scale of tens of milliseconds, remember by 80 milliseconds, we saw no synaptic change. Now tau y seems to operate over a longer time scale, something like hundreds of milliseconds. And this basically tells us that we have a learning rule based on spikes. So we don't have to use the burst rule, but we have a learning rule, the triplet STDP rule, that effectively gives us an elongation of the timescales because we've introduced a third spike that gives us this dependence, this second long timescale that operates on the order of hundreds of milliseconds. So that's how this rule is different from the pairwise STDP rule. And if I now compare this new rule to the data that I showed you before, you see that it matches the data a lot better. There's still a discrepancy here for some of the negative timing experiments, but in general, the rule does pretty well for matching the potentiation. So the cases where the delta T between the pairs of spikes was 10 milliseconds. So what can this rule do? So this is the last bit that I'm going to show you, and then I will summarize everything. So this rule, what I actually showed in my work in 2011 is it can achieve selectivity in a feed-forward network in a very similar way to the BCM rule. Remember, we showed the BCM rule, and we said if we show different orientation patterns, then we basically get the emergence of selectivity in the postsynaptic neuron. But we discussed quite extensively the biological viability of the BCM rule. We looked at this curve that they identified, and we weren't really convinced that this is a rule that really operates in the sensory system, even though it has all these powerful computational properties. We're not so sure if it's relevant biologically. But now we started with the triplet rule by motivating it biologically. So we know it's can explain a lot of data. And so what I was able to achieve in my previous work was to actually demonstrate that this rule, this triplet STDP rule that is biologically motivated, that can explain a large amount of biological data, can be matched very nicely to the BCM rule. So it acquires all of the properties, the computational properties that Venus de Cooper and Monroe argued the rule has. I'm not going to show you the math, but I was able to decompose the change in the synaptic weight into two terms. One term that I call the BCM term, shown here in red, that basically shows us the same kind of dependence, this nonlinear dependence on the postsynaptic activity nu, as in the classical BCM rule. But this triplet rule can do even more than the BCM rule. Now, the BCM rule was purely rate-based. It means that it only cares about firing rates, changes in firing rates to evoke potentiation or depression. But the triplet rule is a spike-based rule. So of course, using spikes, you can get to firing rates because you just count numbers of spikes in a given time window, and that gives you the rate. But spikes give you something that's more than rates. With spikes, you can also calculate correlations. And so it's natural that this triplet rule, because you can reduce spikes to rates, is equivalent or can do the same things as the BCM rule, because BCM rule is just rate-based, and here we have spikes that can be reduced to rates, so therefore can do the same thing as the rate-based rule. But because with spikes you can also calculate correlations, we have this additional term in the triplet rule that allows us to basically generate selectivity to patterns that are also sensitive to correlations. So this is very, very important, and it's a property that we have used also in our recent work. We had a paper in PNAS last year, Wu et al. 2020, where we exactly used the selectivity of the triplet rule to correlations to also generate selectivity in the network to correlations. Now we can, without going into the details, we can demonstrate how this is similar to the BCM rule. We can show that there's a similar type of sliding threshold, and we can have a very nice one-to-one mapping of this mapping threshold, this sliding threshold in the BCM rule into the sliding threshold in the triplet rule. You can look at, there's a hopefully clear appendix in the paper that goes through the math. So what the rule can achieve is selectivity to these dynamic patterns where the pattern basically changes in time. So it's sort of like, I haven't illustrated here, but it's basically showing each of the different orientations at these different time points, T1, T2, T3. And ultimately the goal is selectivity in the postsynaptic neuron to only one of the patterns, even though you show different patterns at different times, where only a subset of the connections that correspond to that one pattern become potentiated. So I'm going to show you just one more detail from my paper. So the way to implement these patterns is to take your postsynaptic neuron and just elevate the firing rate to the synaptic inputs that correspond to that pattern. So here we have 5 weights, 1 through 5. Each one is representing a given, let's say, orientation. And so whenever we present each pattern, it's like we're elevating the rate to that particular input. So for example, at the first time point, we show pattern 5 because we elevate the firing rate to the input 5. Then we show the next time point, we elevate the rate to input 1, then to input 3. So we effectively show each of these different patterns, let's say they correspond to different orientation selectivities, by just elevating the firing rates. And what we see is the gradual strengthening of time over time of one of the synaptic connections. So we can look at the time is here on the x-axis, and we can look at the input number. This is just a schematic, but in this particular time figure of the weights, I show you that each of the inputs is actually represented by a subset. Each of the input patterns is represented by a subset of inputs. So we see the strengthening shown here by the increase in the color from blue to red of a selected pattern. So only one of the patterns corresponding to a given orientation becomes selective because it strengthens in time. But in this case, this is exactly what the BCM rule can do because we're only elevating the firing rates. Now, instead of elevating the firing rates, we can keep the firing rates exactly the same in the network. But what we can do is we can just introduce correlations. So now pattern 3 is not elevated firing rates in these inputs, but it's actually elevated correlations indicated here by these red spikes that are lined up. This is just the schematic. Then the next time point we show pattern 2, where the rates again are the same across all the inputs, but what is different is the elevated correlations. Then we again show pattern 2, because the patterns are chosen randomly, then pattern 1. And so each time we select a given pattern, we just elevate the correlation in the set of inputs that code for that pattern rather than changing the rate. The rate stays here the same. So we still achieve selectivity. So as a function of time, we can look at the strengthening of the synaptic connections that correspond to the different inputs. And just like we saw before, we see gradually the synaptic connection go from blue to red. So the neuron does become selective to one of the patterns. And this particular selectivity property is very different than what the BCM rule can do. The BCM rule cannot do this because here, when we present the patterns, we're not changing the rates, we're just changing the correlations. And the BCM rule is not sensitive to correlations. Only this triplet rule is sensitive to correlations.
So a question, it's probably a little bit fast here. So the correlation is the presynaptic correlations, right?
Yes, exactly.
Okay. But in the rule, right? The rule itself, it's
Repost.
It's postsynaptic, right? So
yeah. So
can you give us some intuition about this, right? Let's go back to the equations that you map your rules with the BCM. Can you give?
We're doing exactly this is a great question. So I'm going to go back way before the beginning of the lecture, because that's exactly what we had at the beginning of the lecture, where I was talking about rate-based plasticity. Let me just go back. It's exactly this point. Remember here, let's say here we also had a learning rule that was defined according to Hebb, where we had the product of the pre and the postsynaptic activity. But we made an assumption about the postsynaptic activity. In this case, we took a so-called linear model and neuron where we had the leak term, and then we were driving the postsynaptic activity by the product of the weights and the presynaptic activity, right? And by doing this separation of time scales, we could write the postsynaptic activity in the steady state and then plug it in to the weight equation. And therefore, we got these correlation and covariance-based rules that purely depended on the correlation and the covariance matrices of the inputs, because we assume this model for the linear neuron, right? So if we now, okay, so I hope you guys remember this, the W dot equal to CW or to QW. So if we now go to our, let me go back to the equation of the rule. So the triplet rule is effectively a combination of the pair-based rule and, let me show you this, yeah. So the triplet rule is effectively a combination of the pair-based rule and this new addition of the triplet. So when we look at what the rule looks like, look at the structure. So we have this BCM term, and then we have this correlation contributions. Look at the first term in the correlation contributions. Looks exactly like the covariance rule, right? We have W dot, okay, I don't have a time constant here because I have absorbed it in the other parameters. But remember, the correlation or the covariance-based rule was a model where I assumed a linear postsynaptic neuron, and so I could plug it in, derive the covariance rule, and the covariance rule was W dot equal to CW or QW. So I had a matrix Q or C, which was exactly the correlation or the covariance matrix of the inputs. So I was able to kind of eliminate the postsynaptic activity by plugging back the model for the postsynaptic activity. So then I just got a dependence.
This delta A is the covariance matrix of the presynaptic neurons?
Exactly. Okay. So this delta A is the pairwise covariance matrix of the presynaptic inputs with some minor modifications, but it's exactly the same thing as the Q and the C that we saw before with the rate-based plasticity. Exactly. And now, because we also have dependence on the third order correlations, we have this other term that looks a little bit convoluted. And the reason that it looks like this is that, so a delta A is a pairwise correlation matrix. And so I can write this easily, right? Because the pairwise correlation matrix, it just means that I can compute correlations between pairs of inputs. I can take input 1 and input 2 and compute the correlation 1, 2, and I put that in the 1, 2 entry in the matrix. But now that I start saying that third order correlations matter, I basically need to have a tensor. So what's a tensor? A tensor is a three-dimensional matrix. So now I need a matrix just like delta A or C or Q that were 2 by 2, sorry, not 2 by 2, but were two-dimensional. I now need to have a tensors because I need to correlate not just inputs 1 and 2, but I need to correlate inputs 1, 2, and 3, and 1, 2, and 4, and 1, 2, and 5. I need to correlate all possible triplet combinations of the inputs. And that gives rise to a third order matrix or so-called tensor that I call delta B. So delta B is three-dimensional. It's like a cube of numbers. Mm-hmm. And I have here chosen to use the index J. So now delta Bj is just a normal matrix. So when I choose to use the index J, it's sort of like I'm taking slices through my cube of my third order tensor of third order correlations. But otherwise, this second part here is supposed to basically indicate kind of mathematically the same thing as this first part. This first part arises because of the pairwise inputs, input correlations.
This W transpose times delta B times W is a scalar, right? So it's 1 times N times N times N times N times 1 matrix, right? So this is a scalar from, right? And the EJ
this is a scalar, and then E is a vector. This is like the, yeah.
I see. And
so
J is J from 1 to N is the number of possibilities, right?
Exactly.
Okay.
That's the number of the inputs that we have. Exactly.
Okay.
Yeah.
So the question I have is, furthermore, because we want people, the students could ask questions as well. So but if you if you don't have triplets, would you still have this BCM term, or you were completely, because normalization can also be done with STDP, the PAL protocol, right? You don't need triplet to do this, right? So
sorry, what kind of normalization?
meaning that the normalization you talk at the very beginning, that
Yes.
cannot go unbounded, right? So
Yes, absolutely.
Yeah.
Yes, absolutely. Normalization can also be done. Let me see. I had actually some slides that I don't know if I can unhide them like this. Maybe I can, maybe I cannot. So normalization can also be done. No, I can show them here. Normalization can also be done in STDP. So if we take the classical pair-based STDP, it suffers from the same problems as the correlation and covariance rules, in the sense that things can grow out of bound. But we can apply separate normalization schemes. So one normalization scheme we can basically set upper bounds on the weights, and we can also impose the subtractive and divisive normalization. So here's an example. I think I already mentioned this paper, Song et al., Song and Abbott, Larry Abbott's the last author, in Neuron, I think, or Nature, where they take this feed-forward network, apply STDP to the weights, and in addition, apply this subtractive normalization.
Sam will be your moderator for the next session.
Yeah. Okay, great. So this will be, so you see that the synaptic weights become extremely competitive. So you basically get they become completely segregated. So about half of them, or some proportion of them, become 0, and the other proportion of them basically reach the upper bound, which in this case is 1. So we have pairwise STDP plus this subtractive normalization that I described to you, that was analyzed for the rate-based rules by Mackay and Miller. And we see kind of the same phenomenon, right? We see that the synaptic weights either become completely depressed or reach what I call the selective fixed point. So we get a scenario like this, right? So we have a scenario, of course, there we have more weights. Here we just have 2. So it's sort of like one of the weight went to 0 and the other weight went to 1. So we pick either this or this fixed point. It's sort of like, here we have 2 weights, and if we build a histogram of 2 weights, we'll see 2 bars, 1 at 0 and 1 at 1, right? That's how it is. That's effectively what I showed you in that plot, right? Let me go back again. Yeah. So that's the pairwise STDP with the subtractive normalization. You can also apply multiplicative normalization. So this is an old study from Mark van Rossum and Gina Julijana and Sasha Nelson, where they actually showed, or they argued, that only weak synapses, sorry, let me see which ones, sorry, the potentiation, they argued that potentiation undergoes multiplicative. The depression is independent of synaptic strength, but potentiation undergoes multiplicative normalization. So for when synapses are being potentiated, then strong and weak ones will experience normalization that's proportional to the synaptic strength. So exactly like this multiplicative normalization that Mackay and Miller studied in their rate-based framework. So remember, for the Mackay and Miller result, they actually showed that if you apply this multiplicative normalization, what you will get is just these so-called non-selective fixed points. So here, both W1 and W2 become non-zero. One is a little bit bigger than the other, but they basically achieve this so-called graded receptive field, meaning that neither weight goes to 0, both of them become non-zero. If we apply this divisive or so-called multiplicative normalization, and that's exactly also what we see here when this was applied together with the STDP rule by Van Rossum. They saw this distribution in the model, this graded distribution. So again, in the phase plane I showed you before, we just had 2 weights. So one was bigger than the other, but both were key, both were non-zero. Here we have many, many weights, and you see that again, none of them go to 0. They just acquire this unimodal distribution. So this kind of gradedness rather than this segregation, half of them go to 0, half of them reach the upper bound. And they argued that this is what they measure experimentally. This was in the visual cortex. So yes, you can indeed add normalization to the classical pair-based STDP rule and still get weights that don't explode or don't decay. But I sort of decided to skip and just show you the matching, the introduction of the triplet rule and the matching to the BCM rule as an alternative of kind of a
it's great. It just probably we can.
Absolutely. A very, very good question. What is important though is, I don't know if you guys want to know more about this, but how this, there's a little trick. So remember the BCM rule has this sliding threshold. So it actually has a threshold that can slide in time. So something, we had this theta equation, differential equation for theta. And so this in the rate-based case can be implemented to sort of sense the postsynaptic activity. The way we do this in the triplet model is we assume that this depression magnitude, originally I called it A minus in the STDP rule. Here I call it A2 minus, 2 because it refers to the kind of pair part. We make this be sliding. So we assume that this A2 minus, so this maximum amount of depression that you can get when the timing delta t is equal to 0 is not constant, but it's sliding. So we have exactly the same dependence on the postsynaptic activity and the target firing rate. So remember, we had a target firing rate and we had a question of how do you choose the target firing rate? So, in some sense, this triplet rule with the sliding threshold will suffer from the same issue as the BCM rule in terms of how you choose this postsynaptic firing rate. But by assuming this kind of sliding amplitude of the depression, you can achieve exactly the same sliding shape of the changing the weight as a function of the postsynaptic activity as in the BCM rule. So by having the triplet rule, you can add this sliding term in the BCM rule, and you can derive exactly what the threshold will be and how it will slide. But then we also have these correlation contributions. So the first component that's very much like the covariance or correlation rule, because it's only the pair-based correlations of the inputs delta A matrix. And then we have this other component that has this basically cubes, this three-dimensional tensor of input correlations. And we did this also analytically, and to make this whole analytics work and this whole mapping, we had to assume the same model for the neuron that I showed you at the very beginning, namely this linear neuron, and we had to do this in the steady state. What we're very interested in doing in the future is to study this for nonlinear neurons, because of course neurons are nonlinear, right? At best, neurons are thresholded, so we can't have negative membrane potential. But here we're kind of working on the limit that we're in a linear neuron scenario, and the neuronal activity cannot actually become negative, and we don't have to worry too much about rectification. So I can hear my baby crying. So I think I would like to make to finish, if that's okay.
Yeah, it's totally fine. Yeah.
Okay.
I don't know if students have other questions. Yeah.
Okay, I'll just finish and then we can go to the
Please just
okay.
Stop here. Yeah.
I just want to end with telling you the classical problem with so we said why we introduced the triplet model, and because it can do the self-normalization that the pair rule cannot do. But the important thing is also that the classical pair rule cannot achieve bidirectional connections unless you do tricks and change the shape of the rule and so on. In other words, the way HEB is defined is you always potentiate the connection from A to B, but you can never simultaneously potentiate the connection from B to A because of the causality, right? The rule is asymmetric, and the triplet rule can actually do this. And so I'm just going to very briefly show you. So we had a paper at some point last year where we showed that the triplet rule can actually do this in a recurrent network. So we can modulate some aspects of the learning rule, and then we can acquire the emergence of these bidirectional connections. So if A potentiates to B, B also potentiates to A. And this is interesting because it allows the formation of these so-called assemblies. So I mentioned assemblies in my abstract. And so the triplet rule can very naturally form these assemblies where groups of neurons become very strongly self-connected to each other. We can visualize these sort of using these connectivity graph motifs. So that's it. I want to end here. This is my map at the end. I showed you rate-based rules, the covariance and the BCM rule. I showed you spike-based rules, the pair and the triplet rule. Basically, the covariance rule can be mapped to the pair, or the pairs TDP rule can do what the covariance rule does and more because of the sensitivity to correlations. The triplet rule can do what the BCM rule does, but also more because of the sensitivity to correlations. And you can make the models more and more complex. You might be familiar with the Klopath rule from 2010, where they use voltage. So you can really kind of build your learning rules, and you can assume, you can have calcium rules as well. As we also show rules that depend on bursts. Most of the rules that I showed you were so-called phenomenological rules because it didn't go into the biophysical implementation of really like what underlies, the potentiation or depression, what are the molecules. But I'm hoping that on in Friday's lecture, I will explain a little bit more of biophysical mechanisms that can implement these rules. And I'll just end with, showing you lots of open issues that we're working on and other people are working on. So inhibitory plasticity is a way to generate EI balance. Everything I showed you was purely excitatory plasticity. Plasticity in recurrent connections. We have started to address this. I showed you one of our recent works, but this is really a big open question about what is the minimal set of mechanisms so you get activity that doesn't blow up or die down. Unfortunately, most of the application has been in kind of sensory systems where we look at like the emergence of simple receptive fields, but how this applies to computations and more interesting memory paradigms that like real animals use is also a big open question. Natural spike trains, the role of neuromodulators. So these are all big open fields really that we and others are working on. So I'd like to just, if there are any burning questions, please ask me. And thank you for the attention.
Yeah, thank you. Yeah, any questions from the audience, please?
Okay, there were questions during the lecture, so hopefully things are clear. I went a little bit fast at the end, but yeah, I mean, I know that one cannot cover all the math aspects, so hopefully you followed mostly the first parts of the lecture, and then you can go into details.
Hi, Professor. I have a question. So I'm wondering, is there any higher function that the local plasticity rule can realize? You talked about the orientation selectivity, this function in this talk. So what's the most complicated function this network or these models can accomplish?
I think it's hard to say what's the most complicated task they could accomplish, but I think it's fair to say, as I wrote in these open issues points, that yeah, most of them have been challenged in the context of evoking very simple sensory plasticity. So simple selectivity in the cortex, like in auditory cortex, neurons become selective to given frequency, in the visual cortex to given orientation. But yeah, these rules are only just now starting to be challenged in more difficult tasks like memory formation. How can you encode multiple memories, and how can you, store and recall? So people are starting to do this, but yeah, it's difficult to say what's the most complicated task they can do, because really they haven't been challenged in so many interesting ways in the past. Only now people are beginning to do this. And maybe some of you work on these things. Yeah, this is open.
Oh yeah, thank you. And could you please recommend some papers on the other functions that it can accomplish?
Yeah, I mean, the people who are working on these actively, so Nicola Brunel is working on some ideas for how these types of models can be used. I think they had some recent paper with Sukbin Lim on, for example, how you acquire senses, how you transition from being selective to kind of novel stimuli, to how eventually you learn when stimuli become familiar. Claudia Klopath has also done some work, especially in Tim Vogel's, in the context of inhibitory plasticity and imprinting of different memories and hippocampus and so on. Yeah, we have also done a little bit of work on what this would mean for maintaining EI balance in sensory cortices, and also for, like, we have used inhibitory plasticity for the generation of novelty responses in the visual cortex based on some experiments with Michael Berry. We have a bioRxiv paper and a paper in review on that at the moment. So if you have trouble locating some of the references based on the names, let me know and email me, and I can forward you the specific references.
Oh, one thing we can do, we can invite you to our Slack channel. Maybe if you have time, you can maybe type in some references.
Sure, I can provide that. Sounds good.
Thank you very much.
Yeah, thank you.
Any other questions?
Okay.
All
then
right. All right.
Thanks for the
Thanks,
attention.
Julijana, again. Okay.
Yeah.
Great.
Okay. Have a nice afternoon.
Yeah. So, so one more thing is we have this live and panel discussion, and there somehow maybe you can. Do you think you will have some time? Maybe in one afternoon. I think from 4 to 4:40. I think.
Maybe we can write an email and we can decide what.
Yeah, we send you email sometime.
Okay.
Yeah, that
Okay.
email. So
Okay,
there are
okay, I
other
can
faculties. Yeah, if you can let me
Okay.
know which is that day.
Okay.
Okay, if you are okay with that day, you don't need to send me an email. You don't need to respond my email.
Okay. I will look again and let you know. Okay.
Okay, great.
Sounds good. Bye, everybody.
Bye.